Okay, so what is a nucleotide? Recap, this is a slide that you've seen before. This is the chemical structure of a nucleotide, showing you those three different parts that I want you guys to know. Recall that a nucleotide is going to consist of a sugar. I'm really going to focus on DNA for this lecture. So the sugar is called deoxyribose. It is highlighted in blue in this image. Another part of a nucleotide is a phosphate group, one of those functional groups, PO4, 1-phosphate, 4 oxygens. That negative charge, that is a phosphate group, very much a part of DNA. Last but not least, we have different nitrogenous bases. So they can either be A, G, C, T, adenine, guanine, cytosine, or thymine in DNA. Right here I'm showing you thymine, that nitrogenous base. And I want you guys to appreciate that that nitrogenous base, part of the nucleotide, kind of hangs off to the side. Here's the sugar, here's a phosphate attached to the sugar. We're going to call this the backbone of DNA. More on that in a bit. Here I can see it just in color format, kind of the backbone of DNA, holding that large biomolecule together. And appreciate that the base is kind of hanging off to the side here. It's a little bit in the periphery of that backbone. So here's thymine. And this is important because I'm going to teach you guys, and you've already seen how that's going to bond to another base with another section, another strand, I should say, of DNA. So there's availability, an area that can bind to another base here. It kind of hangs off to the side. So that's important. Let's go ahead and look one more time at the chemical structure of a polynucleotide. So now having many monomers together. I love this image because it shows you guys that sugar phosphate backbone, and I can actually see kind of a winding structure here. Call that a helical structure. And highlighted is one strand, so showing you here magnified one strand of DNA, that sugar phosphate backbone occurring in repeated monomers with those nitrogenous bases hanging off to the side. This image is that there's another strand in DNA, human DNA, that's also a helical winding structure. It's not highlighted because they didn't blow it up to subsequent images on the right. But what I want you to appreciate here, that this is also a sugar phosphate backbone, and it's going to be the partner of the previous strand because its bases that hang off to the side bond to the other strand's bases. And there's this coding effect that goes on. A bonds with T, G with C, and so forth. And so a strand, it's going to find its partner strand due to this concept.